I band is bisected by Z disc, whereas A band is bisected by M line. The Z disc is built from cap Z, alpha-actinin, desmin and bimentin, whereas M line contains the protein paramyosin, C protein and M protein. And the precise length of thin filament is determined by one of the template protein, nebulin, whereas the thick filaments are positioned midway between the. Hello everyone, welcome back to my channel. So in my last video, I have discussed regarding the reversible binding of a protein to a ligand. I have also discussed the complementary interactions between proteins and ligands. So in this video, I will discuss about the molecular mechanism of muscle contractions, which is basically protein interaction that is modulated by chemical energy. To perform any functions, organisms move, cells move, even organelles and macromolecules within cells move. And most of these movements arise from the activity of a fascinating class of protein-based molecular motors. And these activities are fueled by chemical energy which is usually derived from ATP. Motor proteins play an important role in muscle contraction, migration of organelles along microtubules, rotation of bacterial flagella, and the movement of some proteins along DNA. So now the question is how proteins translate chemical energy into motions. So in this video we are going to discuss the translation of chemical energy into motions by taking an example of contractile proteins of vertebrate skeletal muscle. In vertebrate skeletal muscle, the contractile force is generated by the interaction of two proteins, and these two proteins are myosin and actin. Together, these two proteins make up more than 80% of the protein mass of muscle. And in skeletal muscle, these two proteins are arranged in filaments that undergo transient interactions and slide past each other to bring about the muscle contraction. The myosin protein of skeletal muscle is the first motor protein identified, and it is responsible for generating the force for muscle contraction. Each myosin molecule is composed of 6 subunits, 2 heavy chains and 4 light chains. Here the figure represents the structure of single myosin molecule, which is made up of 2 heavy chains and 4 light chains. And it is the heavy chains that account for much of the overall structure of myosin molecule. Each of the heavy chains has a globular head domain at its end terminus that contains force generating machinery. And this globular head domain is followed by a very long amino acid sequences that forms an extended coiled structure. And this coiled structure mediates the dimerization of heavy chains. An important point regarding the globular head domain is that it contains a site where ATP is hydrolyzed. Now coming to the light chains of myosin molecule. So in myosin molecule, 2 distinct types of light chains are present. As you can see here, this is the magnified image of this portion of myosin molecule. So here you can see clearly the 2 distinct types of myosin molecules are represented in different colors. And 1 copy of each type is present on each myosin head. Means the 2 light chains bind close to the N-terminal globular head domain of each myosin heavy chain. The long coiled portion of the myosin molecule is called tail, whereas the globular domain of the myosin molecule is called head. The tail of one myosin molecule bundles itself with the tails of many other myosin molecules that resulted in tail-tail interactions. So this kind of tail-tail interactions results in the formation of large bipolar thick filament. And the thick filaments have several myosin heads that oriented in opposite directions at the 2 ends of the thick filament, as you can see here. In these thick filaments, the globular domains project from either end of this structure in a regular stacked array, as you can see here. And each myosin head binds and hydrolyzes ATP. And the energy of ATP hydrolysis is used to work towards the plus end of actin filaments that we will see later in this video. And interestingly, the opposing orientations of heads in the thick filament makes the filament efficient at sliding pairs of oppositely oriented actin filaments past each other. Interestingly, the globular domains of myosin molecule has three important sites. First one is the ATP binding site, second one is the actin binding site, and third one is the site where ATP hydrolysis takes place. When the muscle myosin is treated briefly with the protease trypsin, much of the fibrous tail of myosin molecule is cleaved off. That divides the protein into two components, that is light meromyosin and heavy meromyosin. Light meromyosin contains only the extended coil portion of the myosin molecule, whereas heavy meromyosin contains the globular head domain along with some coil portion of the myosin molecule. So when this heavy meromyosin is cleaved with papain, the myosin head group, which is known as myosin subfragment 1 or S1 fragment, is liberated from the heavy meromyosin. Means cleavage of myosin molecule with trypsin and papain separates the myosin S1 fragment from the tail. Similarly, when muscle myosin is digested by chymotrypsin and papain, the globular head domain is released as an intact fragment called S1 fragment. The S1 fragment alone can generate filament sliding in vitro, suggesting that the motor activity is contained completely within the globular head domain of myosin. Therefore, it is the S1 fragment in the motor domain that makes muscle contraction possible. Now coming to actin. Actin is the second major muscle protein after myosin, and it is abundant in almost all eukaryotic cells. Actin occurs in two forms, that is globular actin and filamentous actin. Globular actin is monomeric form of actin, or it is also known as G-Actin, whereas filamentous actin is polymeric form of actin and it is also known as F-Actin. In muscles, the monomeric actin or G-actin associate to form a long polymer helical structure, which is a right-handed coil, and this helical structure is known as F-actin. And the carefully arranged actin filaments are aligned in a thin filament array. And the thin filament consists of F-actin along with troponin and tropomyosin protein. The filamentous parts of thin filaments assemble as successive monomeric actin molecules or G-actin molecules at to one end of the filament, that is at the plus end of the filament. On addition, each monomer or G-actin binds ATP, then hydrolyzes it to ADP. So every actin molecule in the filament is complex with ADP, and the ATP hydrolysis by the actin functions only in the assembly of the filament, means the ATP hydrolysis does not contribute directly to the energy expended in the muscle contraction. And interestingly, each actin monomer in the thin filament bind tightly and specifically to one myosin head group of thick filament. So in conclusion, thick filament is made up of myosin molecules, whereas thin filament is made up of F-actin along with troponin and tropomyosin protein. So till here we have discussed what myosin, actin, thick filament and thin filament. Now coming to the structure of skeletal muscle. So skeletal muscle consists of parallel bundles of muscle fibers, and each muscle fiber is a single very large multinucleated cells formed from many cells fused together, as you can see here. Each muscle fiber is surrounded by membranous structure which is known as sarcoplasmic reticulum. Each muscle fiber contains approximately 1000 myofibrils, which is basic contractile elements of muscle cells. A myofibril is a cylindrical structure of 1 to 2 micrometer in diameter that is often as long as muscle cells itself. Myofibril consists of long repeated chain of tiny contractile units, and that tiny contractile unit is known as sarcomere. And each sarcomere is about 2.2 micrometer long, which is the region between two Z discs. And it is the sarcomere that gives the vertebrate myofibril its striated appearance. Here the figure represents the detailed structure of sarcomere, which is the region between two consecutive Z discs in the myofibril. Each sarcomere is formed from a miniature precisely ordered array of parallel and partly overlapping thick filaments and thin filaments, as you can see here. The thin filaments are composed of F-actin and associated proteins, that is troponin and tropomyosin. Thin filaments are also known as actin filaments. And actin filaments have two ends, that is plus end and minus end. The plus end of actin filaments are attached to the jet disc at both the ends. As you can see here, at this end, the plus end of the actin filament attaches to this jet disc, whereas at this end, the plus end of the actin filament attaches to this jet disc. Whereas the minus end of the actin filament is capped with tropomyosin, and the capped minus ends of the actin filaments extend in towards the middle of the sarcomere, where they overlap with thick filament, as you can see here. And this is the figure that represents the sliding filament model of muscle contraction. And here the actin is represented in red color, myosin is represented in green color, whereas jet disc is represented in light blue color. So according to sliding filament model of muscle contraction, the actin and myosin in a sarcomere slide past one another. And the sarcomere shortening is caused by the myosin filament sliding past the actin thin filaments with no changes in the length of either thick filament or thin filaments. When muscle fibers are examined under electron microscope, the electron microscope study of muscle fiber reveals that there is an alternating regions of high electron density and low electron density. The high electron density corresponds to A band, whereas low electron density corresponds to I band. The A band and I band arise from the arrangement of thick and thin filaments which are aligned and partially overlapping, as you can see here. The I band is the region of the bundle that contains only the thin filaments, as you can see here, whereas the darker A band includes the region where parallel thick and thin filaments overlap, as you can see here. As you can see here, the I band is bisected by a thin structure, and this thin structure is called jet disc. The jet disc is perpendicular to the actin filament. As the actin filaments have both plus end and minus end, so the actin filaments attaches to the jet disc by their plus end. And the jet disc is built from capsid, alpha-actinin, desmin and vimentin. The jet disc caps the actin filament and preventing depolymerization of actin filaments. Now the question is how the precise length of the actin filament is determined in the sarcomere, and it is the template protein of enormous size which is known as nebulin. So it is the nebulin protein that determines the precise length of each filament in the sarcomere, and the nebule protein consists of a repeating 35 amino acids actin binding motif, and the nebule protein stretches from the jet disc to the minus end of each filament. Therefore, nebule protein acts as a molecular ruler to dictate the length of the actin filament. Now coming to thick filaments. As you can see here, the thick filaments are positioned midway between the jet disc. So the question here is, what factors mediate the positioning of the thick filaments midway between the jet disc? And in this, one of the template protein called titin play a major role. The opposing pairs of titin mediates the positioning of thick filaments midway between the jet disc. And the titin is the largest single polypeptide chain discovered thus far. Then the titin links the thick filaments to the jet disc, as you can see here, and providing additional organization to the overall structure. And titin acts as a molecular spring. A spring-like unfolding and refolding of these domains keeps the thick filaments poised in the middle of the sarcomere and allows the muscle fiber to recover after being overstressed. Now coming to A band. The A band is bisected by a thin line, and the thin line is called M line or M disc. M line is the region of high electron density in the middle of the thick filament, as you can see here, and it contains the protein paramyosin, C protein and M protein. So in conclusion, I band is bisected by jet disc, whereas A band is bisected by M line. The jet disc is built from capsid, alpha-actinin, desmin and vimentin, whereas M-line contains the protein paramyosin, C protein and M protein. And the precise length of thin filament is determined by one of the template protein nebulin, whereas the thick filaments are positioned midway between the jet disc by one of the template protein titin. Means nebulin is associated with actin filament or thin filament, whereas titin is associated with thick filament. So till here we have discussed about the structure of vertebrate skeletal muscle and organization of accessory proteins in the sarcomere, which is a basic functional contractile unit of myofibril. Now coming to the molecular mechanism of muscle contraction. During muscle contraction, actin and myosin interacts with each other, and the interaction between actin and myosin involves weak bonds. So when ATP is not bound to myosin, a phase on the myosin head group binds tightly to the actin filament, as you can see here. But when ATP binds to myosin head group and this ATP is hydrolyzed to ADP and inorganic phosphate, there is a coordinated and cyclic series of conformational changes occurs in which the myosin releases the F-actin subunit and binds another F-actin subunit which is further along this filament. And this coordinated and cyclic series of conformational changes has 4 major steps. So first step is the binding of ATP to one of the myosin head group of the thick filament. So once the ATP binds to myosin head group, a cleft in the myosin molecule opens. The opening of cleft in the myosin molecule disrupts the actin-myosin interaction that resulted in the release of bond actin from the myosin head group, as you can see here. Now the hydrolysis of tightly bound ATP causes the conformational changes in the protein to a high energy state, and the high energy state of protein moves the myosin head group and changes the orientation of myosin head group relative to the actin filament. Myosin then binds weakly to an F-actin subunit closer to the Z disc than the one just released. Now, as the phosphate product of ATP hydrolysis is released from myosin, another conformational changes occurs in which myosin clefts closes, and the closing of myosin cleft strengthens the myosin actin binding. And this is followed quickly by a power stroke, during which the conformation of myosin head returns to the original resting state. And finally, ADP is released to complete the cycle. And each cycle of binding and release propels the myosin head forward in a single direction along the actin filament to a new binding site on the actin filament. As during muscle contraction, the thick filaments actively slide forward past the adjacent thin filaments, not backward. So now the question is, what prevents the thick filament from slipping backward when an individual myosin head releases the actin filament to which actin subunit to which it was bonded? Because there are so many myosin heads in a thick filament, and at any given point some are, which is probably 1% to 3%, are bound to thin filaments, and this is the reason why thin filaments not slipping backward when individual myosin head releases the actin subunit to which it was bonded, and the thick filaments thus actively slide forward past the adjacent thin filament, and this process is coordinated among many sarcomeres in muscle fiber that brings about muscle contraction. Now coming to the regulation of muscle contraction. The interaction between actin and myosin must be regulated so that contraction occurs only in response to appropriate signals from the nervous system. And the regulation is mediated by a complex of 2 proteins, that is tropomyosin and troponin. Tropomyosin binds to the thin filament and blocking the attachment site for the myosin head group, whereas troponin is a calcium binding protein. When muscle is in relaxed state, the 2 proteins tropomyosin and troponin arrange around the actin filaments in a manner to block the binding sites for myosin head group. Tropomyosin is a two-stranded coil of alpha helices, which is similar to that of alpha keratin. And the two-stranded coil of alpha helices of tropomyosin is represented in purple color. It forms head-to-tail polymers that twisted around the two actin chains, as you can see here. Whereas troponin complex is attached to the actin tropomyosin complex at a regular interval of 38.5 nanometer. The troponin complex consists of three subunits, that is troponin I, troponin C and troponin T. Troponin I prevents binding of the myosin head group to actin, whereas troponin C has binding sites for calcium ion, and troponin T links the entire troponin complex to tropomyosin, as you can see here. When the muscle receives a neural signal to initiate contraction, calcium ion is released from the sarcoplasmic reticulum, and the released calcium ion binds to troponin C. And the binding of calcium ion to troponin C causes a conformational changes in troponin C, and the conformational changes in troponin C alters the position of troponin I as well as tropomyosin. So after the release of tropomyosin from the actin filament, the myosin binding site is exposed that allow the muscle contraction. So in conclusion, calcium ion plays an important role in muscle contraction by exposing the myosin binding site on the actin filament. I hope you guys like this video, informative for your CSIR exam. So please do not forget to like, share and subscribe, and hit the bell icon just next to the subscribe button. It just helps us stay in touch. And in my next video, I'll discuss about the reaction catalyst of biological system, that is enzyme. I'll discuss about what are enzymes, what are properties of enzymes, and what are the principles underlying their catalytic power. And finally, I'll discuss about the enzyme kinetics.